And it's my particular pleasure to introduce our speaker today because she's somebody of whom I am very fond, who presents provocative and thought-inciting, I don't know what the right way to put that is data, and I use her data all the time to illustrate points. So Dr. Louise McCullough received her PhD in neuroscience as well as her MD from the University of Connecticut. She continued her training at Johns Hopkins, my alma mater as well, where she completed a neurology residency and a fellowship in cerebrovascular disease and eventually joined the faculty there. Dr. McCullough returned to the University of Connecticut in 2004 to develop programs in stroke care and research, where she attained the rank of professor in the Department of Neurology and in the Department of Neuroscience in the Associated John Dempsey Teaching Hospital. She was also appointed the Director of Stroke Research and Education at Hartford Hospital, one of the largest stroke centers in New England. She relocated to the McGovern Medical School at the University of Texas, and I hear Texas is well represented in the audience today, in 2015, where she is currently. At UT, she is a professor and chair of the Department of Neurology, chief of the Neurology Service at Memorial Hermann Hospital, Texas Medical Center, and co-director of the Michener Neuroscience Institute. Dr. McCullough is well recognized for her work in cerebrovascular disease, including her research identifying sex differences in cell death pathways during stroke. Her laboratory also studies aging and inflammation and how these factors, in addition to social factors, interfere and influence recovery after stroke. She's certified by the American Board of Psychiatry and Neurology and has subspecialty board certification in vascular neurology as well as vascular ultrasound. She has been funded by the National Institutes of Health and the American Heart Association for her research, has published over 150 papers, is a prolific mentor, and recipient of numerous teaching, mentoring, physician, and research excellence awards. And as recently as this past January, she was honored with the NIH Javits Award for Stroke Research. As you may already know, the Javits Award is a 7-year research grant award awarded by the NINDS for their scientists for their superior research and outstanding productivity. Recipients of the Javits Award have a history of exceptional talent, imagination, and preeminent scientific achievement. So for today, we're going to hear about stroke, the leading cause of disability in the United States and increasingly recognized as disproportionately affecting women. Although women experience fewer incidents of stroke during the reproductive phases of life, there's a steep increase in stroke incidence in postmenopausal women, as well as sex differences in stroke recovery. Dr. McCullough's lecture will highlight emerging knowledge on the critical influence of sex and age on the response to stroke, demonstrating the importance of modeling sex differences in the laboratory to better inform clinical trial design and clinical care. Dr. McCullough's research is key to understanding the genetic, hormonal, and social underpinnings of stroke recovery. So please join me in welcoming Dr. Louise McCullough.
Thank you. Thank you very much, and thank you for inviting me to give this talk. I do tend to wander, so I'll try not to do it too much. You guys can hear me okay? Because I think it's streamed, so I don't want to get off the camera. All right, so we're going to start with a little bit of a clinical bent. So the first 10 minutes are going to be a little bit about clinical sex differences, because I think they kind of set the stage. I know we have a very kind of varied audience here, and so I wanted to introduce stroke a little bit too, because I know some of you probably are not stroke neurologists, so even though, you know, I hope you all are, because it's cool. All right, I have no disclosures. So we're going to talk mostly about this type of stroke today. This is an ischemic stroke. So these are CAT scans, if you're not used to looking at imaging. This white area is bone, that's calcium. This white area is not good. This is abnormal. This is an intracerebral hemorrhage. This is becoming an increasing problem in our populations, often related to hypertension, but now the second most common cause is cerebral amyloid angiopathy, which is where amyloid is deposited in the vessels of the brain, makes them friable, and makes them likely to hemorrhage. Huge number of patients are affected, and it increases in incidence as we age. As we have more of our elderly patients with AFib that are on anticoagulants, this is becoming a bigger problem. This is a hypertensive hemorrhage. It's deep. Amyloid-related hemorrhages tend to be lobar, so in the cortical areas. This is a CAT scan of a subarachnoid hemorrhage. So again, white stuff is bad unless it's in bone or calcium. So all this white stuff is abnormal. This is an aneurysm that has ruptured. So instead of the blood being inside the parenchyma or the brain, it's in the subarachnoid space. So you can see it kind of casing around the midbrain here. This is usually from aneurysms. This disease does not really have a sex difference, not that we've seen. Men and women are equally affected by intracerebral hemorrhage. Subarachnoid hemorrhage does have a sex difference, and about 60 to 65%, depending on what studies you look at, it's a more female predominant disease. It's also related to estrogen. We still don't understand exactly why. But what we're going to talk about today is ischemic stroke. So this, you can see, there's no blood. There's a little white stuff here. This is actually calcium in the choroid plexus, in the ventricles. But do you guys see this dark hypodense area? That is the infarct. This stroke is probably 6 to 12 hours old. There's not too much mass effect yet. It's not swelling too bad, but you can see the entire middle cerebral artery territory has been infarcted. So when I see a patient like this come into the ER, we're very limited about what treatments we can offer. This is not a patient that you would reperfuse, meaning give a clot buster or put a catheter in their femoral artery to try and get a clot out, because here this brain is dead, and you would just be reperfusing dead brain, making this into something like this. Now, this is our population. This population differs dramatically on what population you look at. This is the intracerebral hemorrhage population, which is growing, again, because of aging and amyloid. And it really depends what race you're looking at. So in Asia or China, ICHs can represent up to 30% or more of strokes. And that's partly genetic, and that's partly due to risk factors like hypertension. The vast majority of strokes are ischemic, and that's what we're going to talk about today. All right, so I'm going to start with a case, and it's not that interactive being up here, but if you like, feel free to shout out stuff. So this is an 85-year-old female, and she just recently was widowed. She lost her long-term husband. She was found by her daughter in the afternoon, who visits weekly, and spoke to her the day before. So we think she's normal the day before. So the first thing you have to figure out is, if you're worried about a stroke, and I get called from the ER, is she focal? Meaning, stroke does not usually affect everything at once, both sides. Usually one side is weak. The face is droopy on one side. The arm is weak on one side. So that's called focality. So her exam was focal. She shows left-sided weakness, face and arm, more than the leg. She also has a gaze preference, and she has neglect. I think you guys probably know what neglect is, but neglect is when you kind of lose that sense of spatial representation, often seen on the left, because the right hemisphere controls kind of spatial perception. The left hemisphere is where language is. So you can kind of localize where a stroke is. And this is not just like neuroanatomy 101, it's like med school. This is really, really, really important in stroke, and I'll explain why. Because neglect is a cortical sign. So it's cortical. It means that the hemisphere itself is involved. That's a lot of territory of the brain. On the left hemisphere, aphasia, trouble getting words out. So people will say, I, or you say, show me 2 fingers. And they can't do it because they don't understand what language is. That's aphasia. That's left hemisphere. Those are both cortical signs. The cortical sign in the back in the occipital lobe is visual field cuts, okay? Why is that important? Because if you have a cortical sign, do you have a large vessel stroke or a small vessel stroke? Rebecca, you're sitting right there. Large vessel stroke. So middle cerebral artery or carotid, that's what supplies the hemisphere. These people they have high stroke scales. They have significant disability and things on exam. They have usually carotid or middle cerebral artery stroke. So if you see somebody with neglect, you're thinking it's right hemisphere. You're thinking it's a large vessel. So I'm thinking, oh, it's carotid or middle cerebral artery. Why do I care? I care because we are in the era of go out and take out the clot. If you have a large vessel stroke, you can go in through the femoral artery and go suck that clot out. The DAWN trial results are not yet out, but they have been presented, that shows that endovascular therapy, going out and taking the clot out of a large vessel, can be done up to 24 hours after stroke onset if people have a penumbra or tissue at risk, 24 hours. So there's going to be select people that may have a stroke the day before and can still get treated. So that's been a game changer. So first of all, where do we think this stroke is? Do we think it's large vessel? Yes. Do we think it's on the left hemisphere or the right hemisphere? Right hemisphere. So we're thinking there's a vascular problem in the right hemisphere because she has neglect. All right, so this is a CAT scan, and I've helped you out a little bit. This is called the red arrow sign. You may be familiar. So if you look closely at this CAT scan, here's the ventricles here where the CSF is made. You can see this side looks pretty normal. This is like, you know, it looks normal, cortical, sulci, fissures. See here? See how this looks kind of blurry and hypodense? This is a completed infarct. This is a patient that probably would not benefit at all from therapy because they have all these early infarct signs. You can still see the caudate in the basal ganglia, but this whole area looks abnormal. And then you can see here, this is called the dense MCA sign. Right in her right MCA, you can see this long, stringy clot, okay? So that tells me she's got a clot in her MCA, but so did her exam. See, exams, I always tell the med students, see, you can't always rely on scans. Exams are important. So her CT angiogram shows proximal right MCA occlusion at M1, but no significant stenosis in the contralateral carotid, really minimal calcifications from anywhere else. So what is the likely etiology of this 85-year-old woman with this large vessel stroke affecting the hemisphere? It's likely from atrial fibrillation. It's from the heart. It's a cardioembolic stroke. They tend to be large. She doesn't have a lot of other vessel disease. Her other vessels look clean, right? So the time of onset is unknown, so what do I do for this lady? Okay? This is what you do. This is what you do in the era of endovascular. This is software called RAPID, which is what was used in the DAWN trial and several other trials. This is a way of looking at diffusion, perfusion. I know here we're very MRI heavy, but these things take about 3 minutes to get, so they're very useful. And basically, you look at cerebral blood volume, and if the blood volume is low, that tells you that area is infarcted. It's not salvageable. This is, and you compare it with perfusion, which is basically you just give a dye, you know, iodinated dye, and you see how long it takes to get to different areas of brain. If it takes longer to get to the right side of the brain than the left side of the brain, it means there's a blockage there. So you can look at basically a CAT scan diffusion perfusion, and you can say, okay, this stuff is dead, this stuff is underperfused. You can also look at her vessels. You can see right here, there's no flow through the M1, and she has very poor collaterals, so not a lot of blood getting here. But is there anything to save? The answer is no. So that CAT scan showing that early hypodensity is confirmed, that that area is already infarcted. So her etiology, AFib. And this is one of the important cases I wanted to talk about because women do so much worse after stroke than men. But you have to remember, there's a lot of differences in the epidemiology of women. Women are anywhere between 5 to 8 years older than men when they have their first stroke, like 68 versus 73, 74. So they have a lot of different comorbidities. Importantly, many of them have AFib because that's an age-related disease of conduction, right? So if you look here, here's a woman, here's the number of women that have atrial fibrillation as a cause for their stroke versus men. So men tend to have more lacunar disease, more atherosclerosis. Women tend to have more cardioembolic strokes. Well, those cardioembolic strokes are big, right? So who's going to do better here? Here's a CAT scan with somebody with a big right MCA stroke. Now, this one started to swell. You can see the whole brain is shifted over to this side. This is a large vessel middle cerebral artery stroke versus this. This is a little diffusion bright lesion on an MRI in the thalamus. This is a little lacune. So I wonder who's going to do better. So this person's obviously going to do better because this is a tiny little infarct versus this. This person is more likely to be a woman this person is more likely to be a man. So you have to factor that in when you think about epidemiology. I wanted to talk a little bit about AFib because it's becoming increasingly prevalent with our aging population. We don't actually model this that well in the laboratory, but women experience more symptoms than men. They tend to have more chest pain. They tend to have more shortness of breath, but they're less likely to receive rhythm control either with drugs or catheter ablation. This is a great review that just came out a couple months ago in Nature Review Cardiology. They have poorer quality of life, and when they have AFib, they have higher risk of stroke. So there's something about women, maybe it's their coagulation factors or something, they are more likely to have a stroke when they're in AFib than a man. They're less likely to be anticoagulated, so there's a double whammy. They're more likely to have an embolic stroke, and they're also less likely to get appropriate blood thinning agents because people feel they're frail, or they live alone, or they're going to fall, they're going to break their hip, they're going to get a subdural. So, and they're also very underrepresented in clinical trials. This is just a diagram showing the number of men versus the number of men versus the number of women enrolled in clinical trials of anticoagulants. These early trials had very, very few women, mostly because they were enrolling at the VA. So now we're doing better. We're kind of getting almost a half-half here. But you have to be very careful extrapolating data from men to women. And this is something I brought up with Janine last time we were at an advisory committee. This is the SPRINT trial, published in New England Journal of Medicine. This is going to change the way we manage blood pressure. This is, oh yeah, blood pressure used to be good. If your blood pressure is 140/80, you're doing awesome. Turns out, if you use intensive blood pressure lowering, you can reduce heart disease and stroke risk by up to a third. But if you look carefully at the analysis here, here's where intensive treatment is better on this side. Here's men, okay? So intensive treatment is clearly better, 30% risk reduction. In women, yes, it's trending because this is standard treatment better, meaning 140, versus intensive treatment, but you'll see the confidence interval crosses 1. So it wasn't significant. Overall, it was significant, but when you break out women alone, clearly there's a trend, and it probably is going to work. But can I say 100% I should be lowering my elderly women's blood pressure to 120? Turns out there was more complications, like they could fall, they could have hip fractures. They were a little underrepresented, but we have to start thinking about this because we have to develop adaptive trials. But if you're not enrolling enough women, or you need a few more to make significance, this trial should have just kept enrolling. Stopped enrolling men. Okay, we get it. We know the answer. We're pretty sure we know the answer here, but maybe not. All right, so I'm going to give an example now from the lab, and the latter part of the talk will be kind of a lot more sciencey, but I wanted to use a few examples here. This is estrogen, one of the biggest translational failures that we've had, right? In observational trials, women who took estrogen were healthier, they had less strokes, they had less fractures, but there was probably a healthy user effect that the people who were taking estrogen were more likely to go to the doctor, were more likely to be non-smokers and stuff like that. So, but in preclinical models, this is a model of, I think this is a rat, but rat, mouse, whatever, rodent. This is crescent violet stained, and here's the striatum here. And this is a female that has intact gonads, so normal estrogen. You induce a stroke, and this is the damage you see in an intact female. If you take out the ovaries, you can see that is huge. We also knew that given estrogen can protect animals from stroke, including male animals. And we're like, oh, it's okay, it's an anti-inflammatory, it's antioxidant, estrogen is great, so we should give it to women. All right, well, this is now an old trial. This is the Women's Health Initiative, which was a very large trial looking at conjugated equine estrogen versus estrogen plus progesterone if they did not have a hysterectomy. And they enrolled patients and followed them over time. And by 4 years, you can start seeing that women treated with either estrogen or estrogen plus progesterone had significantly higher risk of stroke, higher risk of fatal stroke, higher risk of disabling stroke. So all these observational trials, all the epidemiology, all the preclinical data suggesting it was good, well, then you do the trial and it's not good. It's actually quite bad. So estrogen went, within a year from being the most commonly prescribed medication to being almost never prescribe, because in the American Heart Guidelines, we've recommended that people should not be treated with postmenopausal estrogen, okay, for vascular symptoms. But if you look at this study, the people who were enrolled were an average age of 61 to 63, okay? When's the average age of menopause? 51. So these women had gone without any hormones for 10 years and then got probably a super physiological dose of hormone in probably a diseased endothelium, because quite a few were smokers, hypertensive, diabetic, and guess what? They're procoagulant. So could we have designed this study better? The healthy user effect in the observational trials were people who started on estrogen when? At menopause, because they had menopausal symptoms, so they were given estrogen. Now, if you go back to the lab, if you look here, ignore the males, because estrogen is actually protective in males, whether you give it chronically or you give it acutely. But in females, this is what happens if you take this is an old mouse who's about, I think this one's like 20 months old. Mice become kind of reproductively senescent at about 14 months, but these animals were also overrectomized, so there's no way they have any estrogen. This animal was given an estrogen implant at the age of, I think, 13 or 14 months. Then we gave it a stroke at 20 months. The stroke is small. In an animal who was overrectomized and then given estrogen 2 weeks before its stroke, we actually made them much, much worse. So we actually patterned the Women's Health Initiative clinically, and preclinically, but we didn't take what we knew from animal studies to design the clinical trial. And does this matter? Yeah. So this is a New England Journal paper that came out last year, which is called the ELITE trial, which is the early intervention for estrogen. These now took women closer to menopause and gave them either early or late postmenopausal therapy, placebo, or estrogen. And what you can see here, they used a surrogate marker, carotid intima media thickness, because I told you women often don't have strokes till they're 72. These women were enrolled in their 50s, so they're pretty young. We're going to have to watch this group. But you can see if they were given early postmenopausal estrogen, they had very little progression over 6 years of carotid CIMT, atherosclerosis, marker for atherosclerosis, very little. If they were given late, we actually made them a little bit worse. So it does turn out that if we had looked at the design of the trials, we probably would have said, okay, if we're going to give postmenopausal estrogen therapy for vascular protection, we need to do it at the time of menopause. And then after the Women's Health Initiative came out, I don't want you men to feel left out, they did the same trial with testosterone and gave it to like people who are like 78, and guess what happened to the men? They had more strokes and they had more deaths. So this probably is not a sex-specific effect, this is probably an age effect. Okay, now I just want to briefly mention, this will be very quick, because this is stuff I did 10 or 15 years ago, which is actually very, very interesting, and it's kind of set the stage for a lot of the stuff that our lab does now, because we've been trying for decades now to figure out why this happens. And it took a long time for people to accept it, but now everybody has seen it in multiple different cell types, in neurons, in astrocytes, in spleen, in liver cells, and bone marrow cells. So there is sex differences in vitro to stimuli like serum starvation or oxygen glucose deprivation. So this is just stroke in a dish, and basically you take away oxygen and glucose and you can look and see what their cell viability is. We know that females are protected compared to males. Males die faster. And what's important about this is they don't, and this is very complicated. This took us a long time to figure out, but it turns out they're not just, females are a little bit ischemic resistant, but they die by different ways. So male cells, at least in the lab, and most of the cell lines we've looked at, and primary neurons, seem to be dying in males by this caspase-independent pathway, which is activated by DNA damage, PARP activation, and translocation of AIF, apoptosis-inducing factor, to the nucleus, and that splits up all the DNA. And male cells seem to really predominantly die by this pathway. Female cells seem to die by caspase-dependent pathways. And that's when you release cytochrome C release, and it has a different DNA fragmentation pattern. So why does it matter? Because a lot of the drugs that we've targeted may target one side of the pathway and not the other side of the pathway. And then in vitro, if you give those drugs, if you give a drug that's blocks caspases, it's really beneficial for females. It actually doesn't do much for the males. If you have a drug that blocks AIF or PARP, that works really well for the males. It doesn't work very well for the females. And that's true in vitro, and it's true in animal models. Now, who cares? Does it matter? I don't know. Like, does it matter to a stroke patient? Maybe. We don't know why this happens, but I will tell you it has something to do with this molecule, X-linked inhibitor of apoptosis. So it turns out this is really much more complicated than I thought. I have a collaborator and funding, thank you, from the NINDS on this, looking at chromosomal contributions and hormonal contributions. So we use a lot of very complicated chromosomal models. One's called the 4-core genotype. I'm not really going to talk about it much today, but Art Arnold has been a fantastic collaborator, and we have a collaborator at Jack's Labs, Gareth Howell, who is doing sequencing for us and doing epigenetic screens both in human cells, in stroke patients, and in animal cells. So we'll figure it out. It's just taking a while to figure out which gene targets that make this difference. But, you know, this is a lab finding. Who cares, right? So this is important because you have to look. This is animals treated with a PARP inhibitor, okay, minocycline. Okay, it's an antibiotic, it's anti-inflammatory. It's actually been recently tried, this week's New England Journal, in multiple sclerosis. It seemed to have disease-modifying activity in the first 6 months, but then it was gone. But this is now a TTC staining. This is infarct. This pink stuff is live brain. So it's just a coronal section. So this is the infarct. This is a male treated with minocycline. This is the vehicle. This is the male. So you can see a lot of neuroprotection. The animal looks much better. This is a female treated with minocycline, a PARP inhibitor, kind of that male death. So this is the vehicle. Oh, this is the minocycline. So it made no difference whatsoever. But does it matter? So recently, this is a very small study. I think they're going to repeat it. There wasn't enough in the original minocycline or Minos trial of Dave Hess's, which I hope will get re-enrolling, because I think this is a really important question. They found that in a open label study that the minocycline only benefited male stroke patients. It did not benefit female stroke patients at all. So it probably is some signal that in the lab we're seeing that there's no benefit here. We're also seeing it in clinical populations. So there probably is something to this. But does that mean you should abandon this drug? No, it just means you just got to give it to the right person, which is probably a man, right? All right. So how is this woman, going back to our 85-year-old lady found down, how is she going to do? How's this woman going to recover? Well, she's more likely to be disabled from her stroke than a man. She's more likely to die from her stroke. She's 35% more likely to end up in a skilled nursing facility. She has more likely to have cognitive disability, post-stroke depression. But when you think about the epidemiology, it's like that same thing with the stroke size. You have to be careful because she's more likely to have functional deficits before her stroke. And that is something we do not capture in a lot of our databases. Because if a patient comes in using a walker, we have a scale in neurology that says, you know, how disabled you are. If you use a walker, you have a modified Rankin of 3. You come in with your stroke, and you're a woman, and you have a walker, and now your modified Rankin is 5 because you look really bad, right? You have a lot of deficits, you're bedbound. And then I see that patient back at 3 months, and her modified Rankin is 3. Well, that's a great outcome if I knew that her pre-morbid modified Rankin was 3. So she got better, completely better from her stroke, right? But if I didn't know that she was a 3 to begin with, I would say, wow, she looks really bad. That drug didn't work. So we have to be careful to really take into account pre-stroke function because there's much more likely to be disabled if you're a female. And she's more likely to live alone. So age and other factors do matter to outcome. This is a 1-year estimate and a 5-year estimate. This is a recent study as well, showing the classic gender gap in survival. Here's males, here's females, here's males, here's females. But the next slide is busy, so I apologize. But this is the gender gap. But if you look at outcomes when you don't take into account 1-year mortality and you don't adjust for pre-stroke function, women look like they're like 25 to 30% more likely to die. But if you take into account their premorbid function, their premorbid disability, and age and other factors, not just age, not just stroke severity, but premorbid disability, they actually do a bit better. So we have to be very careful with the epidemiology to understand what we're comparing. It's comparing apples and oranges sometimes. All right, so psychosocial factors are much more kind of impactful in women. Depression and stress are more likely to be reported after stroke, up to 45%. These are correlated with higher serum inflammation markers. Now, social isolation or loneliness, we know, increases the risk of mortality after stroke. It's not just stroke, though. It's cancer, you know, any disease, Alzheimer's, any disease is accelerated and magnified by social isolation. Social isolation is very important. It's got to feel negative. You've got to feel subjectively lonely, like you don't have any contacts. You can live alone and be very functional, and you're not lonely, and you don't see the markers of inflammation, which I think is important when we think about clinical trial design. We can't just say, oh, do you live alone? Let's go to your house. Let's, give you contacts, because that may not help, because some people would find it more stressful to have somebody coming to their house. So I think we have to couple this with a biomarker. So isolation also increases the risk of first stroke, and stroke patients with low social support have increased risk of recurrent stroke. Well, I live alone, I'm lonely, I'm an older woman, but you're more likely to be an older woman. Well, I can't get to the pharmacy, I can't get to Walgreens, I'm not going to be compliant. I don't drive very much anymore, so I can't go get my medicines, okay? So there's so many different factors. 52% of women live alone compared to 29% of men. And that's more because they've likely outlived their children and their spouses. If you have a small stroke and you can't get up 3 steps and you live alone, if you're a woman and you're widowed and there's nobody there, guess where you're going? You're not going home. You're going to a SNF. And that's why the rates of nursing home placement are higher for women, because they've often outlived their spouses. There's a lot of different targets, but like I mentioned, it could be compliance. It could be that I'm not taking my medicine. Well, it's not that. And this is some of the stuff the Javits Award is funding, so thanks for that. Basically, what I find really fascinating, because I thought this was all really hokey, and oh, it's compliance, and they're socially isolated, maybe they're depressed. Turns out these things have a huge biological effect. I can take 2 C57 black mice, little mice, they're exactly the same genetics, same diet, same housing, same everything, and I can separate them for 2 weeks and induce a stroke, and look what happens. This is a pair-housed female that's kept with a pair-housed partner, or sometimes we'll keep them in 3s or 4s, and this could be a male that she's used to being with, or another female, and here's her infarct. If we give a mouse that's isolated, you can see this massive increase in the infarct. So just simply separating, and that has nothing to do with compliance, that has nothing to do with genetics. So there's something fundamental here. We do think it has to do with inflammation, and I'm not going to get too into the microRNA story because it's a little bit dense for today, but this is definitely due to microglial inflammation. Microglia get activated in this when animals are isolated, and this is true in both male and female animals. It's worse in females, so very, very kind of nicely mimicking what we see in clinical populations. Now, this animal is going to do really crummy, right? Not surprisingly, right? They have terrible behavioral deficits. They don't get motor function back. They start getting cognitive decline. They look awful, right? But look, their stroke's huge. Like half their hemisphere's gone. So what we wanted to do then is, well, can we separate out the effects of social isolation from the effects on the infarct itself? So what we did then is we gave a stroke, an induced stroke, to animals after, and then waited 3 days and then separated them. We know in our model that the stroke is pretty mature by 3 days, so we then waited, gave them the stroke, kept them pair-housed, and then 3 days later we separated them. And what we found is, here's an isolated mouse, here's a pair-housed mouse with a with a stroke partner. This is a pair-housed mouse with a healthy partner. And honestly, I'll say this, I really thought the stroke mouse would get eaten by the healthy mouse, but they actually don't. They actually groom it. So there's something like in this affiliative behavior, I think, that we need to pay attention to. But you can see here the percent infarct here, this was at 30 and 90 days, was exactly the same. So waiting till the stroke is mature, separating them, all the stroke size was equivalent. But if you look at these animals, this is the isolated mouse. It just continues to die. 80% are dead by 45 days. That has nothing to do with the stroke. They're not getting septic, they're not getting infected. There's something about being isolated, they just continue to die. They sometimes will lose weight. You just find them dead in their cage. We put implantable monitors in, trying to figure out, is it heart rate irregularity, which it does seem to be a little bit, and they just die. And this is what happens in human populations. And if you take one of these isolated mice and you put it in an interaction chamber, it won't interact with other mice at all. And it gets worse. Over time, they get more and more withdrawn. They go to the corner of the cage, they don't vocalize, they don't interact with the other mice, and they die. So we're trying to figure out exactly why this is. And when you can control for stroke size, we also see that the stroke size is the same. These animals recover well. They start recovering their deficits. This animal here that's with a healthy partner, the healthy partner especially, they actually do well. All right, so talking about this stuff is it's really important, like we talked about, in men and women and estrogen and testosterone, to take into account, are we looking at a young brain or are we looking at an aged brain? So I got very into aging over the past 5 years because I was trying to figure out some of the sex difference stuff. And one of the bigger things in sex differences is gonadal hormone exposure. But then I realized it's not all about hormones. It's about aging itself, and it's about the response to stroke in an aged brain. This is a young person. This is an MRI. Brain looks nice and plump. This is a 78-year-old, and you can see lots of white matter disease, huge amount of atrophy here. This is actually a patient with amyloid angiopathy. So we have to really take into account whether we're talking a young female or an old female. The next thing I wanted to talk about really quickly is a risk factor for stroke that is unique to women, and that's pregnancy. So what does pregnancy do to a woman's physiology? Interestingly, if you've had a preeclamptic pregnancy, meaning pregnancy-induced hypertension, which is severe, you have a 2-fold higher risk of stroke later in life and a 4-fold higher risk of hypertension. So something happens to the female's vessels in a preeclamptic pregnancy. What's really interesting, again, I don't want men to feel left out, the offspring of the preeclamptic pregnancy has a 2-fold risk of stroke and a 4-fold risk of hypertension when they are in their 50s and 60s, if you follow these preeclamptic offspring. So there's something that happens both on the maternal side and the fetal side that's probably epigenetic, we're not quite sure, and that patterns risk for much, much later in life. So we started looking a little bit at pregnancy, and one thing that's unique is this thing called microchimerism. This has been known for some time in clinical populations. If a woman has had a boy offspring, they can find XY chromosomes in the bone marrow, cells that have XY chromosomes. They're fetally derived. So a lot of people have said, well, what's the point of this microchimerism? What are these fetal cells? Are they doing anything? Probably not. There's so few of them. But it's kind of cool that they're there and that they can escape immune evasion for decades and decades and harbor out in the bone marrow. But could this somehow pattern women's risk for stroke or their recovery? Good microchimerism, the hypothesis is these are little stem cells. They race to the rescue. They can, help the mom in times of trouble. That's been shown in some cardiac models that these cells can actually traffic to the heart and repair. The bad microchimerism is, guess who gets more autoimmune disease? Women, right? So is it these Y chromosome genes from these fetal cells, are they causing autoimmunity? So we decided to model this in the lab because that's what we do. I got a trigger finger, sorry. This is a GFP positive male. So this mouse is green, and every cell in it is green. You mate this to a GFP negative female. The offspring are heterozygotes and are half green, have half GFP. So what we can do then is every GFP positive cell found in the mom has got to be fetally derived. So this is fetal microchimerism. So we made this model. We bred the animals anywhere from 2 to 6 pregnancies and compared them to virgin females that had never been mated. And that's a good negative control, so they shouldn't have any GFP, obviously. So what we did is we actually, after their last delivery in the first experiment, we gave them a stroke. We wanted to see what would happen, right? And then we analyzed the blood, the brain, and the bone marrow for GFP-positive cells. And what we found is we found these cells in the brain. This doesn't show up too great. This is autofluorescence, so there's nothing on this panel. There's some blue cells you may not be able to see. That's nuclei. That's a DAPI stain. Here's the GFP cells. So we know it's not autofluorescent. So there were these pockets of GFP-positive cells in the maternal brain after stroke. Okay, here they are. Nice little kind of round cells. We immunophenotype them by flow cytometry. These have a lot of stem cell kind of features. Also in the serum of mice that had multiple pregnancies, and it was every pregnancy was higher, they had higher levels of VEGF. Okay, VEGF. Keep that in mind. So what happens is after 72 hours, we would look at this brain and we found these pockets of GFP cells kind of homing to the infarct, and they got there pretty fast. And they were leaving the bone marrow. So we tracked them from the bone marrow to the brain. And what happens to these cells? A month later, look what happens to them. Here's a GFP-positive cell, and it's co-labeled with lectin, tomato lectin, which is a marker for blood vessels. We think they become blood vessels because of the high VEGF signal. So in the serum of mice, the more pregnancies they've had, the higher their VEGF is, and the more angiogenesis they develop. We now know, looking at BRDU, that the number of pregnancies enhances the number of these cells and these BRDU cells, but you can see they clearly form these blood vessels, and they're functional. All right, so last part, well, not the last part, second to last part, third to last part, I'll just talk all day. But anyway, all right, we're gonna change gears a little bit into neuroimmunology. So I like the fetal microchimerism because that is a cell that can reside in a host for decades, in women, and months and months and months, or the life of a mouse, and isn't identified as foreign, which is very interesting. We've gotten very into immunology, and I can thank Lauren Sansing actually for this, because she brought a lot of flow cytometry and immunology to my lab. But neuroimmunology is really the bidirectional communication between the immune system and the nervous system. And I'm going to talk a very little bit at the end. I'm going to add gut in here. So you guys maybe have heard of the microbiome and the gut-brain axis. Well, there's something to it. The CNS is an immune-privileged site. That's probably not true. Cells traffic in and out of the brain, sampling antigens all the time. That kind of crossing through the blood-brain barrier probably occurs more in an older animal or an older person. Inflammation plays a role in a lot of neurological diseases. So also, we have to start thinking of stroke as a systemic disease, right? Stroke is not just about the brain. Recovery is not just about the brain. So basically, this is a thymus in a sham animal and a stroke animal, because we started to see all these T cell changes, so we wanted to look at the thymus. So you can see this animal, 3 days after stroke, their thymus is completely involuted. This is the spleen. So all these peripheral organs are getting a signal from the brain, and they are dumping out their cells and contracting. But then I started thinking, well, aged people and aged mice, we couldn't find the thymus. Well, duh, because the thymus involutes over time and gets really small because you don't have that many naive T cells anymore. They've all been exposed to an antigen, so they're all memory cells or in peripheral immune organs. They're not in the thymus anymore. So we dug around in old mice looking for the thymus for a while before we figured that out. And then we started looking at humans, and they really have small thymus. So we started looking at the immune cells that are normally present in the brain. So this is a flow cytometry plot. This is young brain. This is aged brain. This is CD11B intermediate. This is microglia, just for some of you that are not that familiar with flow cytometry. This is CD45. Microglia are CD45 intermediate. Peripheral leukocytes are CD45 high. So everything in this box here is a peripheral leukocyte, meaning it comes from the systemic circulation. It's not microglia. And here in young versus aged, you can see how many more peripheral cells are kind of just in the brain. This is not a stroke. This is just sham brain. Now, what we found is the cell composition was much greater, but the composition itself was very different. We found a large number of CD8 positive T cells in the aged brain. So we've called these age-related CD8 T cells. It's really imaginative. But anyway, these are important because these CD8 T cells secrete IL-17. IL-17, so they had adhesion markers that allowed them to get into the brain and stay in the brain. And they also were much more likely, this blue, to have high levels of IL-17. That's important, and I'll tell you why in a sec. All right, so I will get back to that. Age mice, we know, this was also like the sex differences stuff, and sex differences in cell signaling took a long time for people to kind of replicate and believe, but it's true. Young animals have much bigger stroke than aged animals. They have a lot more edema, but these animals recover so much better than these animals. And if you look at autopsy specimens in aged individuals as well, we know when we see somebody with an MCA that's 80, we don't see them herniating. You know, we always said, oh, it's because they have, atrophy and they have more room, but they also have less edema. Anyway, so what we found is that the stroke is paradoxically smaller in aged animals, but these aged animals, when we first started doing this, our survival is much, much better. They just die. They're hard to keep alive. We now know how to do it, so we get about a 20% death. But we almost never see death in our young animals, ever. So what we wanted to do is see what did the aged brain do immunologically? How did it respond to stroke, right? So this is the young sham. Again, these are peripheral leukocytes. These are microglia. These are the normal cells kind of trafficking in and out of the brain. This is a perfused brain, so these are not blood cells. We've got the blood out. And this is a young stroke. They get a huge, massive influx of peripheral leukocytes because they have a stroke and the blood-brain barrier breaks down and all those cells go in. In aged sham, again, you see more here. Aged stroke, you actually see a bit fewer cells going in. But the cells going in a young animal are predominantly monocytes. The cells that are going in an aged animal are primarily neutrophils. Well, what do neutrophils respond to? IL-17. So these T cells that have IL-17 in the brain may be calling in the neutrophils. So they're kind of residing there at sham. They're secreting their IL-17. When they get a injury, they call in IL-17 secreting neutrophils. Neutrophils are important. Why? Because they cause bleeding. And we know bleeding is a risk factor for TPA, and bleeding is more common in aged people. So what we did is we generated some heterochronic bone marrow chimeras, where we took young bone marrow and put it into aged animals. We took aged bone marrow, put it into young animals. You can do these with GFP-positive bone marrow, so you can actually track all these little GFP cells. These are all the peripheral cells that come in after a stroke. Here's a stroke, lots of peripheral cells. And what we found is young bone marrow actually enhanced recovery in aged mouse. So this is still an aged mouse. They have an aged brain. They have aged vessels. Everything about them is aged, but their immune system is young. And what we found is you put an old marrow into an old animal, and we get a typical pretty severe neurological deficit, 3 to 4. If we put young bone marrow in 8 weeks before a stroke or 12 weeks before a stroke, these animals did better. They spent more time on a rotorod. They spent more time hanging on a wire, meaning they had stronger grip strength. So the young bone marrow actually contributed to making them better. The aged bone marrow, interestingly, has something really toxic in it. So our young mice, which never die, started dying. They had really bad deficits. So this is a young animal with young bone marrow control versus old bone marrow given to a young animal. So this is still a young animal, but it started doing horribly. And we found that there was no difference in stroke infarct volume. So they were doing horribly, but we weren't making the stroke worse. So what were we doing? Turns out when you look at these immunophenotype, we had changed this monocyte population. So the Ly6G is a marker for neutrophils. So it's 42% in a young. If we give the old bone marrow, 86% became neutrophils coming into the brain. And again, what do neutrophils do? They secrete MMP, and much more than other cell types, and they call in neutrophils, and they cause hemorrhage. So this is a coronal section of a mouse, and see all these little brown spots? That's hemorrhage. We were able to reverse this hemorrhagic phenotype by using young bone marrow into old animals. So it's something in the immune system, not something in the animal, that's causing the hemorrhages in older animals. We could completely reverse the age-related hemorrhage phenotype by giving them young bone marrow. Okay, last part. This is now we got into the microbiome completely backwards. So the microbiome is very hot. I'm sure some of you have studied it. So this is the commensals that live in our gut, you know, millions of bacteria that probably have lots of impact on our host immunity and on the host. So we kind of came across this about 5 years ago, and again, totally backwards. This is a blood agar plate. That's the way I do things. I always do things backwards. This is an aged stroke animal. This is a young stroke animal. We wanted to figure out, well, why are our aged animals dying? Like, what's wrong with them? Do they have heart attacks? Do they have... Turns out they are getting septic, just like our aged patients. The biggest risk factor for death in a stroke patient is infection. These are mesenteric lymph nodes that have been plated out onto a blood agar plate. And you see all those little things? These are colony-forming units of bacteria. And you see in the aged animal, they have a lot more. Here's a young. They even have some at baseline. But we found after stroke, you get a huge amount of bacterial colonization in the mesenteric lymph nodes. So we sequenced them, and we found that most of the colonies that we found in aged were completely different than the bacteria we found in the young. Completely different. So mesenteric lymph nodes, looked at the colonies, sequenced the colonies, totally different bacteria. Lots more bacteria, also completely different bacteria. Well, where were these bacteria coming from? They're gut-derived. So this is a young animal who's been gavaged with Fitzy. This is the outside of the gut. So you can see there's some Fitzy leakage here. This gut looks pretty intact. The aged gut, it just leaks completely. This is a lumen. This is the mucus barrier. This is a normal mucus barrier that prevents bacteria from getting into the systemic circulation. And these are all the immune cells, and our gut is one of our biggest immune organs. This is the mucosal layer in a young animal, in an aged animal, in a stroke animal. This looks really bad. This has nothing to do with gut injury. This happens hours after a stroke. So you give the brain an injury, hours later, the gut is falling apart. You look here, this is the hypoxic barrier. Here's the lumen. There's a hypoxic barrier that, again, prevents bacteria from coming out into the systemic circulation. It's completely gone after a stroke. So then we said, all right, well, maybe this is why they're dying. We should figure out what these are. So let's figure out what are the bacteria in the gut. So we started sequencing the microbiome. And what we found is the young microbiome and the aged microbiome, the bacterial species in there are completely different. They're much more dysbiotic in the age, meaning they have things that are not so commensal and are borderline pathogenic. They secrete LPS. They activate toll-like receptors and cause an inflammatory response in the gut. And we've shown all that. I'm not going to show it right now. And these are just big ways to look at the biome. In the young microbiome, we see many more Bacteroides and fewer firmicutes. And in the old microbiome, we see a big increase in these firmicutes, which are probably a little bit, some families of these are quite pathogenic. So we wanted to find out, can we transmit the biome, kind of like our bone marrow chimeras? Can we take the biome of a young animal and put it in an aged animal? Yeah, it's pretty simple. You take a fecal sample, and we can sequence the donor sample, and then we give it to the aged animal, or we take a young aged sample and give it to the young animal. And what we find is we can completely convert the biome from a young to an aged. So this is the young donor, and this is the aged donor. And if you give a young, the aged recipient, the young donor, we make it look young. If we give the young recipient aged donor, it starts to look like age. That's just F to B ratio. So we can transmit the biome. And what does that mean? We can influence the behavior. So this is a hang wire test. These animals were given biome transfers 1 month before stroke. We sequenced them every week to make sure the biome was stable, and we found that the donor biome was stable. So a young biome given to an aged animal was stable for over 2 months. So once you put that biome in, it does seem to hold for a while. And before we stroked them, which is a topic for another discussion, is the animals that got the young biome, they started to run faster. They lost a little weight. They did better on cognitive tests, just at baseline. So there was kind of a rejuvenating effect of something in the young biome. What is it? We don't know. Is it a bacteria? Is it a bacterial metabolite? And I have a lot of data on that I'm also not showing. But the things that these bacteria secrete may be the things that are really important. Or is it the metabolite or the bacteria's effect on the host immunity. And so now we have to figure out which step, because you can go out to GNC and buy probiotics, but it's not going to work. So this is all a big new hot fad, right? But you may not just need one bacteria, you may need 7 bacteria. You may need one bacteria that produces mucus, but they won't produce mucus unless there's another bacteria in there activating the bacteria that makes mucus. So it's going to be complicated. But what we found also is after stroke is if we gave a young with young microbiome, so this is our control, if we gave young with biome with stroke, they did pretty well after 7 days. But if we gave a young animal aged microbiome, they did horribly. They did worse. And guess what? They started to bleed in their head a little bit. So this is probably all related because we found higher levels of IL-17. And importantly, translationally, in aged mice, you can make them considerably better. Aged mice with young microbiome had much better, oh, is that my own phone? Just, yeah, figures. Just ignore it. It's nothing too important. Anyway, so that's probably me telling me I need to stop anyway. So aged mice, 7 days after ischemic stroke, did much, much better if they had microbiome. All right, that was it. So I just wanted to acknowledge the people that did most of this work, which is Venu Vena, who's done a tremendous amount of the social isolation stuff. Rodney Ritzel did all the bone marrow chimeras. He did the fetal microchimeric work. That's, I think, coming out in PNAS in the next week or two. Fudong Liu. Yor Grafe is our microbiologist who did a lot of the biome stuff. Dave and Rob have been awesome. They are like great. They're at Baylor College of Medicine. They're new collaborators since I moved to Texas. I was very worried about moving to Texas and losing infrastructure. It's fantastic. There's so much down there. So many people to collaborate that I didn't have here. Monica, who's a superstar, Sarah, Laura, and then these are 3 MD PhD students. And most importantly, I want to thank the NIH, and specifically the NINDS, which has given me all my funding over the years, and the American Heart Association. So I wanted to leave a little time for questions, and I wanted to make this kind of more of an overview topic, so hopefully you guys thought something was interesting.
So let's take some questions. Before the first one, let me ask you a microbiome question.
Yes.
There are mice that don't have a microbiome.
Yes, germ-free mice.
Germ-free mice.
Yes.
Have you tried, obviously you need a system for
right.
growing
So
them.
we do. We actually have a germ-free facility, and this is the collaboration I have with Dave and Bob, because they're Baylor. Baylor has a germ-free facility. The problem is it becomes, unless you guys want to give me more money, prohibitively expensive to age a germ-free mouse. Some people have done it. There was actually a recent, very nicely done paper on this that showed that germ-free mice had longer longevity. So not having any bacteria at all, which isn't that practical, actually lengthen lifespan. But I don't know why that is. And again, we don't know if it's something in the biome or something they secrete. The problem with germ-free mice, as Marco Prince has shown, is they have very abnormal microglia. They're very immature. They don't respond to immune challenges. So there's something about the biome that matures the immune system that's probably necessary.
And what about stroke in those animals?
So they have done one experiment with stroke, and they didn't do it in germ-free. What they did, I think, is I think what they did, if I remember correctly, is they did the stroke in one of those minimally biome mice, which have some biome, but minimally they can do the species, and they showed that they did better. But germ-free mice don't do well when they're given a pathogen. So they didn't really look very long after the stroke, and the concern, of course, is that they're going to get infected and die, right? And certainly their immune system is not well handled to really deal with any challenge. So germ-free mice have their own issues. They have a lot of megacolon, their immune system's weird. So I think you just have to be a little bit cautious. We are aging some germ-free mice now, and we'll do the strokes, and we'll see. Nobody's ever done aged germ-free with anything. So I think we have to do it, but it's going to be a little painful.
Also, you can then add back bacteria and see
one at a time. So I will tell you what we're doing there. We've gone the other way that may or may not be the right way, certainly probably isn't, but by giving antibiotics, clearing the host biome, and then adding back bacteria. So what we're doing is kind of, at first we thought, oh, we're just going to take 3 bacteria and we're going to put them together. So a collaborator at Rice is actually making us these genetically engineered bacteria strains that have a tag, either GFP or RFP, so we can follow them, or that overproduce some of the things that we think are beneficial, butyrate, propionate, and to see if we can put those back. But it turns out we probably should do it the other way, which is where we look at the species of bacteria and the metabolomics, figure out what bacteria is making this metabolite, and then put that back. And what we'll do is put back what we see in the young biome, because the young biome is good. The old biome has something very toxic in it, so we could look at that too, because you could say, okay, well, I can use that translationally by trying to block whatever that toxic thing is. So that's the way we're going to do it, is map the metabolites to the 16S and to a thing called shotgun sequencing, try and get a cocktail. So just today, we gave a cocktail of 5 bacteria, genetically engineered bacteria, in animals that got antibiotics, and we'll see what happens.
All right. Question here and over there.
All right. So you propose therapy with bone marrow and microbiome in stroke patients. So that is much better than, I guess, doing stem cell therapy for these patients. And stem cell therapy does not work for the heart, brain, or retina. So how you are going to titrate with? You could measure the volume of the.
That's a great question. So people have tried to use bone marrow because they probably have some hematopoietic stem cells. You have to be careful, like, do you take it from a host? Do you take it from the patient? Obviously, young bone marrow is probably the way to go. Just like young stem cells are probably better than aged stem cells, you know, just as far as their rejuvenating capacity. I don't know if this would ever be a therapy for stroke, but if we can figure out what the mechanism is, it may be a druggable target. Certainly the microbiome could be a very druggable target. If you can figure out what species of bacteria interact to enhance recovery after stroke. And I will tell you, I didn't mention it at all, but we've also done these same microbiome studies post-stroke. And we found that after stroke, you can give a microbiome cocktail at day 4 post-stroke when their stroke is, again, exactly the same size, and you see a huge benefit on recovery. So what is that? I don't know. I don't know if it's the metabolites that we're finding. I don't know if it's the specific bacteria. We've also found a bunch of different T cell populations changing in the gut and changing in the brain. So there's a long way to go with it before we even think about therapy. My concern is, like, people will start saying, oh, probiotics, and then it won't work and everybody will throw it out. But we really have to kind of refine it more than, oh, I'm just going to take some, you know, proactive or something.
So you think this therapy with the bone marrow will not be gender dependent because of the
I think it will be, and you can't give bone marrow. You can give female bone marrow to a male. You can't give male bone marrow to a female because of the Y antigens. But again, youthful factors probably have something to do with it. So would XX be better than XY? Probably, looking at some of our genetic arrays, but we don't know.
Well, we'd like to see the clinical trial.
Oh, no, it would be so cool to do that, right? But again, you have to be cautious because some of the stuff that's been done with Alzheimer's on young blood and young plasma transfers, so they're now giving, in California, aged patients with Alzheimer's whose brains look like that one I showed you that's all like shriveled and atrophied, and they're giving them young blood, off-label. They don't know what's in the young blood because we don't want to heat kill anything, so they're not sterilizing it. It's unbelievable. And they're doing it offshore. They're doing it in Mexico. They're doing stem cell transplants. Who knows what's going on? So now they've been giving young blood to patients with Alzheimer's and other age-related disease. They're going to get hemochromatosis. They're going to get infections. So again, you have to take a cautious approach to these things. They probably do. There's something rejuvenating in the young blood, young plasma, young stem cells, but you can't just throw it at them because it's pretty dangerous.
Vampires have known that for years.
Exactly. My kids will give me something. They're all going to give me a unit of blood.
Yeah, you pretty much answered all the questions I was going to ask, but I'll just make a comment briefly. You talked about ulceration of the GI tract secondary to trauma. I think this has already been noted by Cushing a long time ago, is Cushing ulcers and things like that. So there's really a historic continuity here.
Huge amount. It goes back hundreds of years, actually. And this isn't just the gut. Like, you can clamp the liver and you can see the blood-brain barrier open. And they've known that, and it's kind of like just died out for 100 years. Nobody was really paying attention to some of the stuff that was even done like in 1907 with bacteria and the gut becoming leaky and these things happening in stroke. But I don't think people paid much attention. And honestly, I didn't go at this from the biome side. I went at it trying to figure out, oh, well, what's different? Why are all these aged animals dying? You know, and then it was very clear that the gut was just totally disrupted, and it's disrupted at baseline. It's not just about stroke. That's why aging is so important. You know, now we do half of our studies aren't in stroke animals anymore. It's just age-related phenotype. If you look at the gut of an aged animal or an aged human, it's pro-inflammatory, the villi are flattened, the mucus layer is less complex, and the gut itself, the commensals in the gut, are more dysbiotic. So there's something about aging, and then I think it just sets the tone for chronic inflammation, and then you give a stroke and wham, you know, game over.
Thank you.
All right. Well, let's thank Dr. McCullough for giving us a really neat perspective on stroke. And let me remind you that there's a reception in the library sponsored by the FAES and an opportunity to talk to Dr. McCullough about your favorite ideas.
Thank you for having me.